Welcome to this demonstration on cell passage, a process also known as cell subculturing. In this segment, we will go through the step-by-step process for passaging both adherent and suspension cells. Before beginning, ensure that your work area is properly sterilized. Wipe down the biosafety cabinet with 70% ethanol, wear gloves, and prepare all necessary materials. Before passaging, check the confluence of your adherent cells under the microscope. Cells should ideally be at 80 to 90% confluence. Overconfluence can lead to cell stress and affect proliferation. For suspension cultures, check the cell density and ensure it remains within the optimal range. To passage adherent cells, begin by carefully aspirating the spent medium without disturbing the cell monolayer. Add PBS to the flask to rinse adherent cells and to remove any residual serum that could inhibit trypsin activity. Remove the PBS. Add trypsin EDTA solution to break cell adhesion and ensure the cells are evenly coated. Incubate for 1 to 3 minutes at 37 degrees Celsius, checking frequently under the microscope to avoid overdigestion, and tap the flask gently to aid the process. Once cells are detached, neutralize the trypsin by adding fresh culture medium containing serum. Transfer the suspension to a conical tube and centrifuge for a few minutes to collect the cells into a pellet. After centrifugation, aspirate the supernatant. Gently resuspend the cell pellet in fresh medium. Count the cells if necessary. Transfer the resuspended cells into new culture flasks with pre-warmed culture medium, ensuring even distribution across the surface. Adjust the seating density according to experimental needs. For suspension cells, gently resuspend the culture by pipetting up and down to ensure homogeneous distribution. Then, transfer the required volume to a new culture flask containing fresh medium according to the appropriate dilution factor. This maintains optimal density for continued growth. Once cells are seated, place the culture flasks in an incubator at 37 degrees Celsius with 5% carbon dioxide. Properly label flasks with the passage number, date, and cell type to maintain accurate records. Now, let's dive into the theoretical principles that underpin cell passage. Several factors influence successful cell passage. The correct passage dilution is cell line specific and depends on factors such as doubling time and intended use of the cells. The split ratio is calculated based on the total volume of media used to resuspend the cell pellet after centrifugation. For example, if 10 milliliters of media is used, the total suspension volume will be 10 milliliters. Depending on the cell line, a researcher might follow these split ratios. Different cell types require specific media and environmental conditions. Examples include human embryonic kidney, 293 cells that are typically maintained in Dulbecco's modified eagle medium, supplemented with 10% fetal bovine serum at 37 degrees Celsius with 5% carbon dioxide. Jurkat cells, a suspension cell line, require RPMI 1640 with 10% fetal bovine serum under the same temperature and carbon dioxide conditions. Primary neurons, on the other hand, thrive in neurobasal medium supplemented with B27, providing essential nutrients for neuronal survival. Chinese hamster ovary cells, widely used in protein production, are cultured in HAMS F12 with 10% fetal bovine serum, ensuring optimal growth conditions for these adherent cells. It is important to note that culture conditions may vary depending on the specific sublines of each cell line and experimental protocols. Therefore, it is recommended to consult the data sheets from cell suppliers or relevant scientific publications to obtain precise information about culture conditions suited to your experimental needs. Cell detachment is facilitated by enzymes like trypsin, which break down cell adhesion proteins. The effectiveness of detachment depends on several factors. First, the trypsin incubation time is crucial, as overexposure can damage cells. Second, the removal of calcium and magnesium is important because these ions stabilize cell adhesion. Therefore, PBS without calcium or magnesium is used before trypsinization. Lastly, Mechanical methods, such as gentle pipetting or using cell scrapers, can help some cell types detach more effectively. Selecting the right culture vessel is crucial for optimal growth. Adherent cells are commonly maintained in T25, T75, or T175 flasks, with the choice depending on the required cell yield. Multi-well plates, available in 6, 12, 24, and 96 well formats, are ideal for high-throughput experiments and drug testing, offering scalability for different research needs. Suspension cultures benefit from spinner flasks, which provide continuous agitation to enhance cell proliferation. For large-scale industrial production, bioreactors are the preferred choice, offering precise environmental control for cell expansion and protein expression. Now that we've covered the theoretical principles, let's review common mistakes to avoid during cell passage. Leaving cells in trypsin for too long can damage surface proteins, leading to poor attachment and reduced viability. Always monitor detachment and neutralize trypsin promptly. Seeding too few or too many cells affect growth and experimental reproducibility. Always optimize density according to your experimental needs. Poor aseptic techniques